the audience. All right. It's really a great honor to host today's seminar today. Introduce our distinguished speaker, Dr. Deborah Burnett, who publishes so well and then has been doing fun research. And Deborah's journey into immunology began with her background in veterinary medicine, which ignited her fascination with the immune system. She then transitioned into medical research through an honor degree at the Walter and Elisa Hall Institute and earned her PhD at the Garvan Institute. When the COVID-19 pandemic emerged, Deborah quickly pivoted her expertise to develop vaccination strategies aimed at inducing antibodies resistant to viral escape. Her groundbreaking discoveries earned her the highly commended Discovery Award from Research Australia in 2022. Deborah is now a Santisha Senior Lecturer at the UNSW School of Biomedical Sciences, where she leads a laboratory focused on developing innovative models to study antibody cross-reactivity in vaccines and autoimmunity. Based on what I learned a minute ago, maybe she's an independent PI. I'm not quite sure how it works, but congratulations as an independent PI with 4 people already. Very nice. Her impressive research portfolio includes publications in top-tier journals such as Science, Cell Immunity, and PNS. In 2023, she was honored as one of 4 Australian um, Oriel UNESCO for Women Science Fellows and received her Premier's Prize for Early Career Research of the Year. Please join me in welcome Dr. Deborah Burnett to share her invaluable insights with us today. Thank you, Deborah.
Thank you. And, just thank you so much. I was just saying beforehand, this is such a fantastic seminar series. I've really enjoyed watching a lot of the previous seminars. And I think it's just a great way for us to share science across the world without having to deal with long haul flights. So I'm really, really grateful for the organizers for inviting me to speak today. And so, yeah, so thank you very much. I'd also like to acknowledge the ancestral owners of the land where my work was conducted, which is the Gadigal people of the Eora Nation. So they're the First Nations people in Australia, and pay my respects to elders past, present, and emerging. So I think Paul actually did a fantastic job of introducing me already, so I don't really need to say that much. Just that actually, as Paul said, I've actually just started my own laboratory as a PI just in November last year, so incredibly recent. It's a really exciting time. So my laboratory is focusing on exploring B cells in vaccines and autoimmune settings. And so today I'm going to be talking about where my research has come from and some of our previous research and sort of branch out into the research we're doing now. And I just want to flag that we do have PhD and master's positions available in my team. So if the research today looks really interesting to you, please reach out. I'm based in Sydney. It's really beautiful, as you can see here. And just one more thing, just to sell it, when I'm not working in the lab, I'm actually a wildlife rehabilitation and carer for orphaned baby Australian animals. They're incredibly cute. So if that doesn't give you enough motivation to come visit our lab in Australia, nothing will. Okay, so now on to the science. So today I'm going to be talking to you about the role of antibodies produced by B cells. So when we think about B cells and antibodies and their function, what we all think about is their main role, which is fighting invading microorganisms and providing protective immunity. But really, in my mind, B cells at all times have 2 main roles. Of course, they have to produce antibodies for fighting infections, but at the same time, their equally important role is to avoid producing antibodies which bind to our own tissues and lead to autoimmunity. And so I think we've got an immunology audience, so I'm going to go into a bit of background here, which I think we all know, but I think it's always helpful to sort of begin, from the same baseline. So I'm going to talk a little bit more about what we all understand, the B cell response to foreign antigens. So there are numerous possible threats to the immune system, And as we all saw in 2020, this includes pathogens which don't even exist yet. So there has to be this system for B cells where there's an enormous plasticity and ability to adapt to new antigens that the immune system has never encountered. So the way they do this is there's a huge variety of B cells, each specific for a particular antigen. And when a B cell encounters its cognate antigen, it becomes activated, it enters germinal centers, it undergoes proliferation, and it undergoes this random mutation of its antibody receptor via somatic hypermutation. Those B cells which have increased their affinity for the foreign antigen are then positively selected. And then, of course, these positively selected B cells are then differentiate into long-lived plasmablasts and leads to the production of antibodies. And so the reason I stress this is because this is what we really sort of understand well, and a lot of groups are studying at the moment, what drives this process of affinity maturation and the mechanisms that underpin this. So what we all know and agree that within the germinal center, B cells acquire mutations in the region encoding their antibody receptor. These mutations are random, but those mutations that increase foreign affinity undergo positive selection, and those mutations that decrease foreign affinity undergo negative selection. And that's all very good, but what about self-reactivity? So the enormous diversity of the B cells that we require to allow us to cover all possible foreign threats does mean that many of these B cells will be cross-reactive between the foreign antigens and the self-antigens. And so this is why it's so important that the immune system has the capacity to balance that risk of autoimmunity versus protection. It's now recognized that actually the majority of B cells produced in the bone marrow by VDJ recombination actually recognize self-antigens. And so the immune system has various mechanisms to mitigate the effects of these self-reactive B cells. Those B cells that have a high affinity for self undergo deletion or death or receptor editing before they even make it out into the periphery. However, those B cells with a low affinity for the self-antigen or even a moderate affinity for the self-antigen do make it out to the periphery. And instead, they're largely controlled by this process of inactivation called anergy. And it's an active process, and it can be reversed. And so this really poses the age-old question. It's well recognized that the self-reactive B cells have the potential to become reactivated, and this is what drives a lot of autoimmune diseases. So the age-old question is, if these self-reactive B cells, if these energic B cells in the periphery have the potential to cause autoimmune disease, why are they being kept alive and suppressed in the first state, rather than just being deleted before they left the bone marrow? So this is the age-old question. And so when we think about age-old questions, here's another age-old question. Why did the chicken cross the road? And of course, this one we know the answer to. The answer to this question is to get to the other side. So we began investigating what if the answer to our B cell age-old question was the same? So if there was permanent inactivation of self-reactive B cells, this would create potential holes in the immune repertoire, which would be conserved across the population. And so these could be exploited by pathogens. A way around this could involve reactivation and rescue of self-reactive clones. So this is the theory of clonal redemption. So for this theory of clonal redemption to be correct, there would be 4 criteria that would need to be fulfilled. These self-reactive clones would need to be rescued from deletion and from their short half-lives, and they would need to be reawakened and enter the germinal center. Within the germinal center, they would have to have the capacity to undergo somatic hypermutation, and this would include somatic hypermutation of their B cell receptor away from self-binding. So they'd have to be able to reduce self-binding for them to be safely reactivated and leave the germinal center without causing autoimmunity. And of course, they would have to undergo the somatic hypermutation process that we all know underpins normal affinity maturation. So they would have to be able to undergo somatic hypermutation of their B cell receptors towards the foreign antigen as well. So to investigate this further, we required a model where we could trace B cells specific for a low or moderate affinity self-antigen. And so to do this, we utilized the switch-held B cell system, which was developed by Chris Goodnow and Rob Brink. And so I'll just go into this. I think many of you are probably familiar with this system already, but for those who aren't, switch-held B cells are antigen-specific B cells expressing the high HEL10 B cell receptor. So throughout this talk, I'm often going to use the term switch-held B cells or high HEL10 B cells interchangeably. And so the high HEL10 B cell receptor has a very high affinity for Hennig lysozyme, or HEL. And so we wanted a system where we can trace B cells specific for a moderate or low affinity self-antigen. So as our self-antigen, we utilized HEL3X. And so this is Hennig lysozyme with 3 amino acid substitutions. And as you can see here, instead of having an incredibly high affinity for the high HEL10 B cells, it has a moderate affinity for high HEL10. And so then we required a mouse model. So we utilized these mice, which were initially established by Rob Brink and Tiani Chan. And so these mice expressed HEL3X as a membrane-bound, ubiquitously expressed self-antigen, so expressed as a surface antigen on all their tissues. And so now we have these antigen-specific B cells in a mouse model where they're expressing the self-antigen that they have moderate affinity for. So to make that, we irradiated these HEL3X transgenic mice and also non-transgenic recipients. And then we reconstituted their bone marrow with a combination of the same bone marrow from the mice we had irradiated and also these high HEL10 expressing B cells. So now we've got these high HEL10 B cells being a small proportion of the bone marrow of these mice. And in the HEL3X transgenic mice, these B cells are self-reactive. And so when we explored these mice 8 weeks after reconstitution, we can see that these high HEL10 expressing switch-held B cells show signs consistent with anergy. So they had a shortened lifetime. You can see here that they had IgM downregulation while maintaining IgD, which is classic phenergic B cells. And they also had this profile where they were excluded from the follicular zone and the marginal zone, and instead were mostly within the T cell zone. So this is really classic signs of clonal anergy. And so now we have this model. We've got high HEL10 specific B cells developing in mice expressing HEL3X as a self-antigen. So these B cells are inert. And so now we can start to investigate this clonal redemption hypothesis. And to do this, we expose the mice to a foreign antigen mimicking self. And so in the initial experiments I'm going to show you, we conjugated all the antigens to foreign erythrocytes, so sheep red blood cells. And this is again it's an artificial, but it's a very well-studied system. And so the sheep red blood cells, the foreign erythrocytes, provide the source of T cell help. And so it's important to recognize in this system the T cells are not self-reactive. And so this system has been very well characterized. And in fact, the mechanism of how this recognition has been published by Jason Sister's group. And we know it's a good immune stimuli. And so for these experiments, we immunized the mice with the foreign antigen mimicking self, and we initially used one of 2 foreign antigens. So in some of these experiments, the foreign antigen was HEL3X. And so this means that the foreign antigen and the self-antigen are identical. And in other experiments, the foreign antigen was duck egg lysozyme, or DEL. And you can see here that DEL has a similar affinity for these high HEL10 expressing B cells as HEL3X. However, it's structurally different. So there is a difference between the foreign antigen and the self-antigen, but in both cases, they have the same affinity for the B cells. So the next thing we wanted to look at is how these B cells, or whether these B cells are recruited into the germinal center. And so what I'm showing here is initially the non-self-reactive B cells. And so this is looking really early in the response. so 4 days following a pseudo-primary immunization. And so we're just looking at that point when they're just being recruited into the germinal center. And what you can see is that both of these foreign antigens are very efficient at recruiting these B cells into the germinal, these antigen-specific B cells into the germinal center. And you can see that these two foreign antigens recruited these B cells in similar proportions, which makes sense because both these B cells have the same, sorry, both these antigens have the same affinity for that B cell receptor. That's all well and good. So what if it's self-reactive? So what you can see, which is really interesting, is that when the switch-L B cells were self-reactive in the HEL3X transgenic mice, they were recruited into the germinal center at the same proportions as if they were non-self-reactive. That's really interesting. And what's also interesting is at this really early time point, it didn't really matter whether or not the foreign antigen was identical to the self antigen in the HEL3X, or if the foreign antigen differed from the self antigen. So what we could see from this is anergic B cells can be efficiently recruited into the germinal center. Just to remind you, this is using high-density antigen conjugated on sheep red blood cells. We've got a source of T cell help. It's been established by previous experiments that this reversal of energy does need a strong source of T cell help. What's really interesting is we saw that they were very efficiently recruited into the germinal center response, but actually they avoided the plasmablast response, which you can see here. And that's really important because it meant that they were safely being recruited, and there was no evidence of a breach intolerance because the germinal center is still a process where there's further controls applied. And they were not recruited into the memory compartment. And so we wanted to understand further what are the factors necessary for this recruitment into the germinal center. And so one of the things we investigated, as I mentioned, this was using really high density foreign antigen. So we wanted to understand how important that high density of the antigen was. And so we conjugated the foreign antigen on the sheep red blood cells, so on our foreign source, at different densities. And so this is here, you can see we conjugated it on the sheep red blood cells at really high density, or we conjugated HEL3X on the sheep red blood cells at a much lower density. And you can see that this lower density that we conjugated was near identical to the density that the membrane-bound HEL3X is endogenously expressed as a self-antigen on the HEL3X transgenic mice. Okay, so first let's remove self-reactivity and just look at the recruitment of these switch-L B cells in a non-self-reactive system to this high and low density antigen. Oops, sorry. So here you can see that this is looking at the non-self-reactive switch-L B cells. And so the high density antigen was very able to recruit these B cells into the germinal center, but the low density antigen was still able to recruit these B cells into the germinal center when they're not self-reactive, although it was admittedly at a lower proportion. So what about when these B cells are self-reactive? You can see that the high density antigen was able to recruit them into the germinal center, but when the when the foreign antigen was at the same density as the self antigen, these switch-L B cells were no longer being recruited into the germinal center at all. So recruitment into the germinal center is dependent on the density of the foreign antigen being higher than that of the self antigen. And so you can see that when we have low density self antigen, even if there's sufficient T cell help, we're no longer seeing it being recruited into any of these compartments. And so actually just to jump to some really new stuff, one of the things that my team is really interested in now is understanding how this actually applies to autoimmunity that follows many viral infections. And so it's well recognized that many infections are associated with an increase in autoimmune disease, but also that there's a transient allowance for self-reactive antibodies, or there's a transient production of self-reactive antibodies following many infectious causes. So this was all using our very safe sheep red blood cell foreign erythrocyte system. And so these are actually some very recent experiments which have been conducted by Adrian, a PhD student within my team. And so this is using a very similar model. I won't go into the details of it, but actually in this case, instead of just immunizing the mice, Adrian has been infecting the mice with a variant of ectromelia virus or mousepox, which expresses HEL3X. And so this is for anyone who's actually done the experiments with mousepox, it's an incredibly severe viral infection. So it's actually lethal in BALB/c mice. These were mice on a black 6 background. So it's not lethal, but it's a pretty strong immune stimuli. The mice definitely develop clinical signs. And interestingly, what we saw in this case, so here you can see this is looking at serum self-binding antibodies. And so here in the top is the non-transgenic mice. So there's no self-reactivity, and you can see that they form lots of serum HEL3X binding antibodies. And this is actually our negative control mice all down the bottom. And here what you can see is actually, this is the HEL3X transgenic mice. And you can see that actually in this case, they are forming, there is a significant increase in self-binding antibodies. So that suggests that if the viral stimuli is strong enough, the immune system will preferentially form antibodies against the foreign antigen, preferentially, even if it results in some form of break of self-tolerance. However, this serum autoreactivity was transient. And so when we look at a later time point, we're still seeing in the non-self-reactive system that there are serum antibodies binding HEL3X, but we're no longer seeing the self-reactive antibodies in the HEL3X transgenic mice. And I will flag that this was just serum antibodies. We didn't actually see any clinical signs caused by these. So just to flag that actually, if we have a strong enough viral stimuli, we do see some recruitment of these B cells also into the plasmablast compartment while they're still self-reactive, which is really interesting. Okay, so that sort of covers what the processes are, well, what's happening with the recruitment of the germinal center B cells. But what about maintenance within the germinal center over time? And so now we know that we've got these self-reactive B cells that can enter the germinal center pretty much in normal frequencies, even when they're self-reactive. And so what happens over time? So now we're looking at actually a much later stage in the response, and this is back to the sheep red blood cell experiments. And so you can see here that when we're looking at non-self-reactive switch cell B cells, so non-transgenic mice, the B cells are retained within the germinal center at similar frequencies from both this HEL3X immunization and this DEL immunization. And again, this makes sense because HEL3X and DEL have similar affinities for the high HEL10 for the B cells. So what about self-reactivity? And so this is when we start to really see the differences in the self-reactive system. So when the self-antigen and the foreign antigen were identical, so we've got HEL3X transgenic mice and we immunize them with HEL3X sheep red blood cells, these B cells were efficiently recruited into the germinal center, but as you can see, they are not maintained within the germinal center over time. However, when the foreign antigen and the self-antigen differed, you can see that these switch cell B cells are actually retained within the germinal center over time. So maintenance within the germinal center requires a path for escape of self-reactivity. And so what if we actually look at these B cells a bit more to understand how this process is occurring in terms of their affinity? And so this is looking in the case of the HEL3X immunization. So the self and the foreign antigen are identical. And you can see that these B cells are, this is looking at their HEL3X binding. And so this is the non-self-reactive switch cell B cells. And so you can see they have lots of binding to HEL3X, which makes sense because it's the foreign antigen. And so we single cell sequence these B cells and we sequence their B cell receptor and then we express the antibodies and determine the effects of the mutations that these B cells had acquired on affinity to the foreign antigen. And as you'd expect, all the mutations they've acquired increase their affinity to the foreign antigen, which is HEL3X. So what about when HEL3X is also the self-antigen? And so these are the B cells within the germinal center. And you can see that even those B cells that are retained within the germinal center, they no longer bind HEL3X, which is the foreign antigen, but it's also the self-antigen. When we sequence these B cells and determined the affinity of the mutations they've acquired in their antibody receptor, we found that they had acquired these mutations that dramatically decrease affinity to HEL3X. So they acquire mutations that decrease affinity to the self-antigen and the foreign antigen. And again, this is really interesting because when we come back to those plots I showed you, well, sorry, the introduction I gave you about how we understand affinity maturation, this goes completely against that. We're seeing processes within the germinal center where B cells are acquiring mutations that decrease affinity towards the foreign antigen. So when the self and foreign antigen are identical, self-reactive B cells will lose self-reactivity, but if they're identical, that also means they're losing foreign reactivity. So what about when the self and foreign antigen differ? So again, this is without DEL immunization, and again, we're starting by looking in the non-self-reactive system. So we've got switch L B cells immunized with DEL, and so this is the affinity effects of the mutations they acquire. And so all the mutations they acquire increase their affinity to DEL, the foreign antigen. Some of the mutations they acquire have increased affinity to HEL3X, some of them have decreased affinity to HEL3X, but that's largely irrelevant because it's not a self-antigen in these mice. However, when HEL3X is a self-antigen, we see a much more selective mutational profile. So they're still acquiring mutations that increase affinity to the foreign antigen, but they're doing so in a selective way so that the only mutations that persist are mutations that concurrently have also, well, B cells that have acquired mutations that have lowered their affinity to the self antigen. So when the self and foreign antigen differ, self-reactive B cells lose self-reactivity and maintain foreign reactivity. So now we can come to the age-old question number 2, which is which came first, the chicken or the egg? So we know we have these B cells that have acquired mutations that have decreased their self-reactivity and also increased their foreign reactivity. But the question is, which of these came first? Did they first acquire mutations to decrease their self-reactivity, or did they first acquire mutations to increase their foreign reactivity? And so to do this, we look specifically only in the HEL3X transgenic mice. And again, we sequenced the B cells and determined the affinity of the mutations they'd acquired in their B cell receptors. But the difference is we did it at several different time points. So initially we looked at this really early time point in the germinal center. And so you can see that these switch L B cells at this time point, the majority of the mutations they have acquired certainly decrease, and these are all mice immunized with duck egg lysozymes. So the foreign antigen and the self antigen are different. And so the mutations they've acquired have decreased their self-binding, but the first mutations they've acquired, once again, actually have also concurrently, they've decreased self-binding, but they've also decreased foreign binding. And so what about when we look a bit later in the response? And so when we looked a bit later in this response, we found that the predominant mutations these B cells were acquiring were still able to decrease self-binding, but they did so in a way that they maintained foreign binding. So there was very little effect of foreign binding of these particular mutations. And so once these B cells had acquired these mutations that decreased self-binding, it was only at that point that they were able to acquire secondary mutations, which then increased their foreign binding, similar in the way that traditional affinity maturation occurs. So this means that we now know that actually the first process that occurs is this reduction of self-binding. And initially it happens in a very non-specific way, followed by more specific mutations, which decrease affinity to self while maintaining foreign. And then we get an increase in foreign affinity. So that's all very well and good. We've confirmed how the immune response responds to hen egg lysozyme. What is the physiological relevance of this? And so really this is actually work not by my team, but by other groups worldwide. And so there's now been quite a few different publications where other groups have confirmed that this clonal redemption process is part of the normal pathogen response to lots of different pathogens. And the key point being that when this process happens, normally you wouldn't even know, because the only time that we actually see these B cells emerging from the germinal center is when they've already lost their self-reactivity. So this is actually lots of different pathogens, many of which we don't actually think of as pathogens which mimic self because the immune system has always been able to overcome this challenge with the B cells acquiring mutations to discriminate self from foreign. Interestingly, there are teams worldwide exploring whether clonal redemption of B cells is involved in the response to cancer. And I think that's really interesting, and we'll watch this space and see how that develops. So I'm just going to quickly specifically highlight the clonal redemption in the case of vaccinia. So this is some work by Joanne Reed. And so she was looking at VH434 antibodies that had been previously characterized by Iñaki Sanz's group as part of the normal response to vaccinia. And so these antibodies were not self-reactive and they were just known to be part of the vaccinia response. And then when Joanne Reed reverted them to their pre-immune form prior to somatic hypermutations, she found that they actually had significant self-reactivity. And the reason why I flagged that in particular is because VH434 is a great example of this paradigm of self-reactive B cells between protective immunity and autoimmunity. So VH434 cells are known to be involved in many normal pathogen responses, but this same antibody isotype is well recognized to drive many autoimmune diseases. So VH434 antibodies are predominantly driving cold agglutinin disease, and they're also significantly enriched in lupus. And so this is really the direction that my team is now investigating. We're trying to understand the mechanism of clonal redemption. And this is really important for several reasons. The first is vaccine development. So, in many cases, the B cells are able to undergo clonal redemption and be redeemed, and we don't even need to think about it because the immune system can do it on its own. However, there's various examples of pathogens where the immune system has not been able to successfully uncover a pathway for redemption, things like HIV. And so understanding how this process of clonal redemption works and how we could manipulate it may allow us to be better able to enhance B cell vaccine development against pathogens which highly mimic self, and there hasn't been a clear pathway for vaccine development yet. And so we might need to reactivate self-reactive B cells. The other reason it's really important is because these same cells are involved in autoimmune disease. So if we understand the normal mechanisms that are involved in this process, we can suggest gene pathways that could be investigated as potentially driving these autoimmune diseases. So for the rest of the talk, I'm going to talk about very, very recent unpublished work that we're doing to understand the mechanisms of clonal redemption. So we wanted to establish first a molecular basis for how these reawakened B cells are recruited into the germinal center. So we've got self-reactive B cells entering the germinal center, and how does this process differ from non-self-reactive B cells being recruited into the germinal center? And so to do this, we looked at this really early germinal center response. Initially, when the self-reactive B cells, really early. And so at this time point, when we see the same numbers of B cells being recruited, whether they're self-reactive or not self-reactive. And so we sequenced these antigen-specific B cells and performed RNA-seq. And what you can see here is that there is a clear molecular signature which completely separates these antigen-specific B cells with the same specificity compared to B cells with the same specificity, but in a non-self-reactive system. What's also quite interesting on this PCA is you can see that in the non-self-reactive system, there's much less variability between different mice. Whereas even though there is still a consistent gene signature with over 1,000 genes which are consistently differentially regulated, there's much more variability in this self-reactive system. And this may relate to the challenges that these B cells are seeing as they're at different stages of testing this early time point. They're really uncovering these pathways for redemption and which ones allow those B cells to survive. And I'll come back to that a bit more. So now we had a molecular signature for these reawakened B cells, and we wanted to compare this to previously published molecular signatures. So the first molecular signature we wanted to compare it to was the gene signature associated with anergy. So as I mentioned, anergy is a process that applies to naive, self-reactive but naive B cells. And now we've got self-reactive germinal center B cells. And so what's the similarities? And so sure enough, we found that there was a significant association with this anergy signature of naive B cells with the self-reactive B cell signature in the germinal center. However, what was interesting, it's not the entire signature that's upregulated or downregulated. There's actually just a subset of anergy genes which are consistently associated with this self-reactive germinal center signature. The next thing we wanted to compare it to was the gene signature, which has been previously published within the germinal center to be associated with T cell help. And so interestingly, what we found is this anergy gene, sorry, this is not anergy, it's this reawakened germinal center GC signature was actually associated with a loss of T cell help, which is really interesting. However, if you remember, when we're talking about this really early time point, the majority of the mutations that the B cells are requiring at this time point are mutations that decrease self-reactivity and foreign reactivity. So what this highlighted to us is actually we need to go a bit deeper. We can't just look just at this reawakened B cell signature because there's just too much variability. We've got some B cells that have lost T cell help, and we've got some that are actually beginning to specifically acquire mutations while T cell help is maintained. So we need to not just look at the reawakened gene cell signature, but we need to establish this molecular signature which underpins this reduction in self-binding and this completely unknown process, which goes against everything we know about affinity maturation. And so to do this, we looked specifically from the HEL3X transgenic mice. And so we separated them into those B cells that have retained their binding to the self-antigen and those that have already acquired mutations to lose the self-antigen. And we did this by single-cell RNA sequencing, and we've done several different experiments, and as I'll show you at different time points. And in some experiments, we separated the binding from non-binding just by their flow profile of binding, flow cytometry binding to the self-antigen. But in later experiments, we actually combined this with 10x VDJ. And so we could look at the RNA profile specifically in those cells that had acquired specific mutations. Okay, so this is showing the data from if we remember at that really early time point, this is what redemption looks like. We're losing self-reactivity and foreign reactivity at the same time. Whereas when we go to a later time point, redemption is now a process where we lose self-reactivity, but maintain foreign reactivity. The RNA signature of self-reactivity at this early time point is really, really distinct. So those redeemed cells that have actually lost their self-binding pretty much completely separate from those cells that still retain self-binding. However, when we look at this later time point, the signature is much more subtle. And we're no longer seeing that effects caused by the loss of T cell help, which is actually the predominant factor driving a lot of these. And so this is really useful by having these multiple time points and in addition, those experiments where we combine it with VDJ, because now we could start to ask specifically what aspects of the redemption signature are not related to their binding to the foreign antigen, but are specifically associated with that process of redemption across multiple time points, regardless of what it's doing to foreign affinity. And so sure enough, when we look at these multiple experiments, we only see, instead of having thousands of genes which are upregulated or downregulated, we now have a very small subset of genes which are consistently associated with redemption across multiple experiments. And unfortunately, just because it's a recorded presentation and this is still, I can't quite, for IP reasons, I can't quite go into the exact genes that we're investigating, but now we have a consistent molecular signature. And so the experiments that we're doing at the moment are to determine which of these are just associated and which of these are mechanistic. And if you are interested in knowing more, please do contact me because I am happy to talk anyone through these. I just can't quite announce them in this presentation, but it shouldn't be too far before we can share that data. So bringing this together, reawakened B cells within the germinal center share a gene signature which is associated with that naive state of energy. And redeemed cells actually have a distinct molecular signature, which is quite unlike anything we've seen before. And there's a couple of really exciting gene subset or genes that we're investigating as potentially driving this process. Okay, so let's just conclude with this fine balance of immune control. So the immune system really is walking on a delicate balance. And so it has the choice of actually keeping all self-binding B cells alive and active, but this would lead them to attack self-antigens and cause autoimmunity. Alternatively, the immune system could take an approach where it deleted all B cells binding self-antigens, even within the bone marrow, but this would lead us unable to fight pathogens mimicking self and vulnerable to infections. So instead, what we see is a process whereby B cells that are moderately self-reactive are not deleted, and instead they're kept suppressed, but in this active state of energy. And this active state means that they're ready to be reawakened and mutate away from self-reactivity to combat pathogens mimicking self. And this is what underpins protective immunity to pathogens mimicking self. So this is obviously a work of a huge number of years, really my career to date. And so I'd like to thank a number of people. So a lot of this work was done, in fact, most of it was done when I was in Chris Goodnow's lab. And so I really want to thank a lot of people in Chris Goodnow's lab. And in particular, I want to flag Tim Peters, who is still the bioinformatician who's driving a huge part of the molecular signature work that's still ongoing. Rob Brink and Tiani Chan were essential for establishing many of the models that we were using in these experiments. And Tim Newson from University of Sydney developed the ectromelia virus. And finally, I'd like to thank people in my current lab, and in particular flagging Adrian Chi, who performed those ectromelia experiments. And so, yes, thank you. Is there any questions?
Thanks, Deborah. It's fantastic and so exciting research, and to me, it's really brought me into the new conceptual ideas of the B cell biology. I'm not very familiar with the B cell biology. It seems very exciting, and then understanding how it works in different regulations. So now let's just see if we have a questions from the audience. If you have any questions, please raise your hand, or I can, or we can just communicate with your questions with Deborah directly. Or we can leave your question in the chat box, and then we'll read it out for you as well. So maybe I just take advantage of the chance to ask some questions. I'm not, again, very familiar with the B cells, but this is so exciting. So the question I have in my mind, just maybe I also can learn the knowledge from you, is that there's some antigens, for example, the lipids. I'm coming from the lipid
Mm.
biology. So the lipids can be recognized by, for example, iNKT cell, and then communicate with B cell to produce antibody. And in this context can be important in atherosclerosis, for example, the cardiovascular disease. So, what, to my knowledge, maybe the lipid cannot serve as antigen to interact with B cell directly, right? How does that work? Any ideas?
That's a great question. And it's really interesting. So, I mean, the work that I've just shown you is predominantly using proteins mimicking self.
Right, right.
So we have had to certainly
Mm,
carbohydrates mimicking self are important in a lot of settings. And so, we know that, for example, HIV is coated in self-glycans, and same with LCMV. And so that side of things definitely has been investigated. And similarly, we've got cases like autoimmune disease where in GBS, where the terminal oligosaccharide is near identical on human nerves.
mhm,
But I
mhm.
genuinely haven't considered lipids. And I think that's really, I don't have an answer to it. And I think it'd be a really interesting area to investigate. I'd be very happy to follow up with you after if you can tell me of some
Mhm.
examples where we know that there's foreign lipids mimicking self, because I don't know whether or not it would be the same. And it'd be really interesting to investigate.
Yeah, yeah. This is probably just another a challenging layer of the antigen presentation and antigen biology in B cell. So following the question, like nowadays people also study immune metabolism and then try to tease apart the metabolism inside the B cell and then how it affects those responses, right? That it should show
Mm.
the energy, this and that. Can you comment on the different context? I'm gonna assume that what the mice that you are using is all chow fed, right? Like for example, if mouse have
Yes.
if a metabolism background like diabetes or high lipids background, or how will that shift what it just showed us, and then how
It's a
uh,
it's a great question as well. So, all the work we've done so far has been in black six mice as well.
right.
So apart from everything else, we're using a mouse model which is not predisposed
Mm-hmm.
to autoimmunity. And so,
Mm-hmm.
as I said, when we use the sheep red blood cell system, we have this system where the B cells are incredibly safely recruited into the germinal center, and they
Right.
only leave when they've lost their self-reactivity.
Mm-hmm.
And it's a really interesting point of how do we gear this system towards autoimmunity? Because obviously that does occur following a lot of infections. And so I briefly
Mm-hmm.
shared that data of if you have a strong enough viral infection, you can get a transient autoimmunity. But there's a lot of other things that we could investigate. So something, as you say, we could try change the diet. We could try this in obese mice. I think even just doing this in a different mouse strain would have a huge effect. What we have done already is we have done some microbiome work where we co-house the mice with dirty microbiome mice. And it's really interesting because in our hands in some cases that did exacerbate an autoimmune phenotype. So like A20, we find that A20 mice normally we didn't get an autoimmune phenotype, but when we had dirty microbiome, they did. But that did not exacerbate this autoantibody redemption process. But it is really interesting sort of understanding to how much genetic, environmental, dietary would change that seesaw balance more on the
Right.
side of the autoimmunity. Because
Yes.
obviously we know that in some people we are seeing autoimmune diseases following infections. And so what I'm really interested in that space and what's driving that.
Yeah, I see. Yeah, so exciting. So this, what your study can apply to, different contexts, including, for example, just aging, right? Aging is also popular too. Um,
Yeah, true. So we have definitely done it in older mice, and that we haven't yet. So aging, we haven't seen it still works in the older mice, and in fact,
mhm.
often actually, we haven't done it in young mice. I'd say probably because one of the things is with this chimeric system, you irradiate the mice, and the minimum amount of time. So then you have to wait 8 weeks post reconstitution, and actually, so by definition, we've done it in sort of normal, well, 20-week-old mice to heading up to a year old and we haven't seen a difference, but we've never done it in really young mice. And so that could be really interesting too.
Awesome, awesome. So we have a question from chat box, from B Xingyun. Curious about that if a patient with a severe autoimmune disease like SLE got infected by some bacteria or virus that contains a foreign antigen, would the autoreactive B cells turn the BCR affinity to the foreign antigen instead of the self antigen? Um,
Yeah, so that's really interesting. So it is known
mhm,
uh, in HIV that there are some published works. I actually can't remember the author, but there is, I've got a review which has the references to the published work on it, if you want to look that up, that in HIV, patients with SLE are more able to form broadly neutralizing antibodies to HIV. So in people who potentially might be genetically predisposed to autoimmunity, who are exposed to a foreign antigen that mimics self, that there is not an easy pathway for redemption, there is evidence that those patients are more able to form those antibodies. Now, interestingly, what I didn't talk too much about in this talk is one of the things we've been very interested in is this process and what about cancer immunotherapy? Because that's an example where we can actually, people are given agents, anti-CTLA-4 and anti-PD-1, to predispose them to autoimmunity. And so Adrian Chi, the PhD student, one of the things he's really been looking at is actually how this process is affected in immunotherapy. And interestingly, we found that it's not. There's well-published evidence that we can agree with that there's an overall expansion of B cells, but this germinal center tolerance mechanism is not affected by anti-CTLA-4 and anti-PD-1, which is really interesting.
Hmm. Okay, so I think he has another follow-up questions. Do you think toll-like receptor will be involved in the process? I mean, this is probably related to the question he asked.
Yeah, I mean, it's a good question. I don't have an answer. Yeah, it very much could be.
Mhm. Okay. Is there any more questions? If there's no more questions, let's thanks Dr. Deborah Burnett again. And this is a wonderful talk. And if I am sure that if you have any questions, if you contact Deborah, I mean, you can develop a further communication and then learn with each other.
Thank you
Um,
so much again.
yeah, thank you, Deborah. And then hopefully I can meet you in person someday. And
Yes, please.
welcome to Shanghai, China. If you have any chance, please contact me. I will write an
Thank
email to you.
Yes, please. Please do. Thank you so much.
Okay, thanks so much. Yeah, have a nice day.